Hello, this is Miss Dagenford, and I wanted to give us a little bit of a head start for next week. We're going to be talking about the biotechnology that can be done and giving a little bit of taste for some of the procedures that are out there. Now, we already did our virtual lab on polymerase chain reaction, so you have a little bit of a sense of some of the stuff that's out there. The fact that we can just copy and amplify DNA, which is just pretty crazy. And so one of the first things I wanted to talk about was just, well, what can we do with some of the DNA that we might have? Well, one of the first things we might do is create a recombinant piece of DNA and clone it. And now you hear cloning, and that's sometimes a kind of a crazy idea that pops into your head. But cloning can just simply be creating a bacteria that will be making a certain protein for us. And in simple terms, what we do is we create a bacteria that has a gene that we want it to have, and it's going to be making a protein for us. Now, how does this work? Well, let's make sure we recall some facts about bacteria. So over here is a picture of bacteria, and bacteria have one big circular piece of DNA. They don't have, they have one long circular chromosome. They don't have linear chromosomes like eukaryotic cells do. They have one loop. But one thing they do have are these little circular pieces of DNA called plasmids, and these are just small pieces. They might have maybe um, just a few hundred nucleotides, just enough for maybe three, five, 7 genes on them. These aren't big pieces of DNA, but they can be very, very useful. And they're small enough that bacteria can absorb them from outside. So here would be a little plasmid, and if we create the right conditions, the bacteria will happily absorb the plasmid from outside and through its cell walls. Now, what kind of plasmid would we want to make? Well, if we have a gene that we like, that we want to make a lot of that protein, well, bacteria will happily do that as long as you give it the gene. And so over on the picture on the right, you might see the eukaryotic cell that has a gene that we want. Now, the only problem is this is a eukaryotic cell. So genes would have exons and introns and other exons. And if we were to give that gene to a bacteria, the bacteria wouldn't know what to do with the exons. It doesn't splice, it doesn't have a spliceosome. And so it would make a junk protein because you've told it to make those introns. So what we can do is we can actually we want a mature mRNA. And I say mature because those are the ones that have already been processed. The introns have been taken out. And what we can do is we can take that mRNA and use an enzyme called reverse transcriptase to make a cDNA, a copy of that DNA. And that's oftentimes what gets put back into the plasmid, is the copy of that mature mRNA. Then once it's in the bacteria, the bacteria is just going to make whatever protein we want. And we can do this lots and lots and lots of different ways and for different reasons. We can make medicines this way. We can make transgenic corn. And in fact, some of you had to do a biotech project back in your freshman year for this. So some of you already have some familiarity with this. So now, how do we do this? Well, let's talk about an enzyme that we're going to be familiar with called restriction enzymes. And restriction enzymes, just as enzymes are, they have a specific active site, and that active site is going to fit on a certain sequence of DNA. And these restriction enzymes are actually protective. They're found in bacteria, and the idea is that they are going to cut DNA from viruses. When a virus attacks a bacteria, these restriction enzymes will fit into the piece of DNA that has that specific sequence and cut. Now it's going to cut the backbone, that sugar phosphate backbone, and it's going to cut in a very unique way. It's going to cut on the between the G and the C on the upper strand, and then on the lower strand, same thing. And notice this is the same on the top and the bottom, but it's backwards. It's palindrome. Palindromic. It's a palindrome. GAATTC CTTAAG. And so if it cuts between the G and the A, it's going to look like the second picture. Now, there's something unique about this. Because it's asymmetrical, that leaves nucleotides free to make a hydrogen bond. So these nucleotides that are available, that they can now make a hydrogen bond with any DNA that has its sequence complementary to it. So if we saw an AATT, then it would hydrogen bond. Well, what if we took a gene that was also cut with this restriction enzyme? Well, lo and behold, you would get a sticky end that fits right in these little places. And that's what we can do. Now, the only key is you have to use the same restriction enzyme. There are lots of different restriction enzymes. They're named after the bacteria that they're found in. The one I just showed you is called EcoR1 from Escherichia coli, restriction 1. There's a Hindi 3 that's well known. There's a BAM, and these are all from different bacteria. But the key is you're only going to get these lovely sticky ends coming together if you use the same restriction enzyme. And we're going to do a little exercise to kind of help us visualize that. And then the hydrogen bonds happily develop, but that's not enough to keep your gene there. So you use DNA ligase, and DNA ligase is going to connect that backbone up, and then you'll have a secure piece of DNA. Now, the plasmid that we are going to use this week is a plasmid that has been engineered for us. The company makes it, and it makes a plasmid with a gene that ends up glowing, and the plasmid's called pGlow. And the gene that they use is a gene from a jellyfish, and it's called green fluorescent protein. So this gene is from a jellyfish, and we're putting that jellyfish gene into, well, they've put it into a plasmid, and then we're going to induce the bacteria to take it up, and that's called transformation. And the reason they were able to do that is because they used restriction enzymes to create a sticky end, and then they were able to put the gene that they want. Now, the cool thing about their plasmid is they've also attached a promoter that is actually part of an operon. And that means we get to turn it on and off based upon the conditions we put it in. And so we'll be learning about that and reading about that early next week. So just kind of to sum up, we reviewed bacteria a little bit, the two DNA structures in bacteria, the main bacterial chromosome, and then the small circular plasmids, which we're going to be looking at. We can engineer plasmids and insert genes into them pretty logically and easily by using restriction enzymes, using the same restriction enzymes, so we get a clean and consistent sticky end with a gene that we want, and then by using DNA ligase to sew it up. If you don't use ligase, it's not secure. So that's one use of restriction enzymes, and that will help us get started next week. So thank you, and have a good weekend.